In our last unit, we spent quite a bit of time talking about meiosis and the process of separating chromosomes and the alleles that those chromosomes are encoding into different gametes. Because in meiosis, we go from a diploid stem cell to haploid gametes. Right? And that's important because at fertilization, we want two haploid gametes to come back together and restore the diploid condition. So what we're going to do in these next few videos is very briefly introduce you to some of the basic principles of inheritance, of how genetic information is being inherited and the patterns of that inheritance. And the purpose is not to be completely exhaustive, but for this class, it's to get you ready for taking your next course, which is an upper division genetics class that most of you will be taking in the fall. So let's start with a basic definition. Genetics is the scientific study of heredity, inheritance, and variation in that inheritance. So what might that look like? Here's an old picture of my family from many years ago. Boy, this is probably a dozen years old. And there's me in the middle, and my half-sister to my right, and our dad to her right. And then, I guess I'm saying that wrong, right, from where we're standing. But then over here is my wife, Ann, and she's holding our youngest son, Josh, and here's our two other kids, Ryan and Sydney. And so there's all kinds of interesting relationships among these people. And genetics is really all about understanding how the DNA and the phenotypes that the DNA ultimately produces are being passed from one generation to the next. That's really what we're trying to understand here. Gregor Mendel was a monk who is famous for having done a bunch of initial genetics work with flowers and peas. And so we're going to start off by looking at some of Gregor Mendel's work and some of the basics of this. If you've seen it before in another class, great. Just view this as review. But what I'm really hoping to do is help you tie this in with what goes on during meiosis, because that connection is not always clear, and I want to make sure you understand that. A couple of terms we've used earlier when we were talking about meiosis specifically is the idea of a character and a trait. And a character in genetics refers to some feature, like petal color, that can be inherited from one generation to the next. Trait defines which variants of the character. So red petals would be a trait. White petals would be a trait. So we're going to be tracking characters and traits as we move through these different essentially mating experiments. And we're going to work with plants primarily. The other thing we're going to do is we're going to focus, for the sake of illustration, on characters that are readily visible, understanding that the majority of the characters that are being inherited aren't visible. They're happening at an invisible biochemical level. But it's so much easier to wrap our brains around this for the first time by thinking about things like the height of a plant. So here was how Gregor Mendel looked at some of his pea plants. If you look at the top of the page, we've got the pea generation. P stands for parental generation. And what he did in his parental generation was he had tall plants that were what he called pure breeding, meaning that he could cross them with each other and cross them with themselves, and the only thing they ever gave were more tall plants. And then he also had a population of plants that were pure breeding dwarf, meaning he'd cross them with themselves, got nothing but dwarf plants, he'd cross them with their siblings, got nothing but dwarf plants. They were pure breeding. There appeared to be no information for anything other than how to be a dwarf plant. So he took a pure breeding tall plant and crossed it with a pure breeding dwarf plant. And he did this many times over. And what he got was all tall offspring. And there weren't just 4. There were hundreds and hundreds of these. Now this first generation we call the F1, or first filial generation. So we've got our parental generation that we know are pure breeding tall and pure breeding dwarf. We mate them, we cross them, and we get 100% tall offspring. What happened to the genetics of this poor little dwarf guy? Well, we'll be able to find that out and tease that apart here in just a minute. The next thing he did was he took a couple of his F1 generation and either self-fertilized them, which plants can do, or cross-fertilized just within the F1 generation. And he got something different. He got 25% roughly of his F2 generation as being dwarf, showing that phenotype that was absent in the F1 generation. So in the F1 generation, that dwarf phenotype seemed to just disappear. F2 generation, it reemerged at approximately 25% of the offspring, and the other 75% were tall. Now let's add a little modern genotypic information to what Mendel was looking at with just plain phenotypes. It's really pretty remarkable how much he was able to figure out without understanding modern genetics. A pure breeding tall plant is going to be represented with two large letter T's. Now, what these large T's represent are the genotype with regards to this particular character, nothing else, right? All the other genes are still present. We're just tracking this one character right now. But the genotype, meaning the versions or alleles of this particular gene that's determining plant height, that are present in that parent. Now we know that that parent, that organism, that plant, is diploid. And so there has to be two copies. And in this case, there was one copy it received from one parent, one copy it received from the other parent, and it happened to both be the same copy. It happened to be an allele for the character of height that coded for the trait tall. They got a tall allele and a second tall allele. No wonder when these guys were self-crossed and crossed with siblings, they were pure breeding. You got nothing but tall, because the only DNA present are tall genes. Now, when our dwarf, pure breeding dwarf, meant we had little t, meaning we had another allele for the plant height gene, and it was an allele that coded for a dwarf phenotype, the phenotype being the expression of the gene, the measurable, visible expression of the gene. And you can, again, see why when it's crossbred and when it's self-fertilized, you got nothing but dwarf plants, because the only genes available are dwarf genes. There are no tall genes. And so he didn't understand all this, but he was able to elucidate some pretty incredible principles just based on phenotype. So when a pure breeding tall plant is crossed with a pure breeding dwarf plant, right? Because of the way that meiosis works, only one or the other gametes, or one or the other, allele gets put into each gamete. So in this case, the pure breeding tall plant, all of its gametes had the capital T tall allele. And the pure breeding dwarf plant, all of its gametes had the lowercase t dwarf allele. And so when those gametes fertilized, our result was what we call a heterozygote. Heterozygote means that it has one of the, in this case, tall allele and one of the dwarf allele. So now we've got 50-50. Now, another principle that Mendel was able to elucidate here was this idea of dominance and recessiveness. And it turned out that with this particular trait, if the dominant allele was present in at least one copy, then the plants were going to have the dominant phenotype. In other words, the big T tall allele was dominant over the little t dwarf allele, and therefore the dwarf allele was never expressed. The tall allele was expressed, and 100% of the F1 generation showed the tall phenotype. Makes sense. So that's the idea of being heterozygous. So we crossed a homozygous dominant tall parent with a homozygous recessive dwarf parent, and we ended up with heterozygous tall F1 offspring. But now when these offspring are either self-fertilized or fertilized amongst each other, each gamete gets either the big T allele or the little t allele, but not both, because we know that in meiosis we separate the gametes out because we reduce the ploidy from diploid to haploid. And so half the gametes would have the big T, half would have the little t, and then if we cross that with another heterozygote, half would have big T, half would have little t. And what he ended up seeing was 3 out of every 4 offspring, 75% of them, having the tall phenotype. So either big T, or big T, little t. So either homozygous dominant or heterozygous. In either case, you're going to have the dominant phenotype. And 1 out of 4 had the recessive phenotype because 1 out of 4 got both little t alleles from both parents. So hopefully that makes sense. If it doesn't, think it through. But let's take a look at it from slightly different perspective. So here's our F1 generation, our heterozygotes being crossed. Remember, the plant itself is diploid, but its gametes are going to be haploid, meaning they'll carry one allele or the other, but not both alleles in the gametes. And therefore, if half of this plant's gametes carry the big T and half carry the little t, and the same for this one, then we've got the opportunity for big T to fertilize a big T, or big T could fertilize little t, and then vice versa, little t fertilizing the big T and little t fertilizing little t, leading us to one big T homozygous dominant for every 2 heterozygous big T little t genotypes for every, again, 1 homozygous recessive. And what we see phenotypically is 3 to 1, tall to short. And genotypically, we see 1 to 2 to 1, homozygous dominant to heterozygous to homozygous recessive. And this all comes back to the process of segregation of alleles during meiosis, and then essentially recombining, if you will, of these alleles back in fertilization again. And we're going to talk about this principle of segregation in the next short video. But in the meantime, I want you to spend a little time thinking about this one. Make sure it makes sense before you go on to the next video.